We continue. So the proteins such as the trypsin, and the pepsin that you learned before. Trypsin is secreted out from the pancreas. Pepsin is from the stomach itself. They will help to break down or digest, or you can say digest, up to you, the egg stain. So they will digest the proteins into the amino acid. Yeah. So you say that enzyme. So this enzyme, you should mention the specific enzyme, which is the protease and trypsin and pepsin. So they react with the protein substrate and digest it. Okay. This one you should say that after they digest the protein will become amino acids. So proteins will digested to become amino acid, which is their simplest form of the monomer for the proteins. So, and then you see, they give you 3 marks. So you should write at least 3 words. So you should write that enzyme. What enzyme will do? Enzyme able to enzyme, or you can say that enzyme protease. They able to carry out the chemical digestions, because only enzyme able to carry out the chemical digestion, right? Chemical digestions. Okay, that's all. So the mark will be given to here, then amino acid, then chemical digestion. So those are the 3 marks. Okay, so if you write like this, maybe you can care about 1 mark. Okay, at least you write the at least you write about digest proteins. And then, okay, this one. Why the washing powder, they work best at 30 degrees Celsius and they should not be used above 50 degrees Celsius. So the reason is, you say that yes, the optimum is about 30 degrees Celsius. Okay, you can get about 1 mark. Then if the temperature beyond the 50 degrees Celsius, it will denature the enzyme. Yes, good, correct, you write it. And then this will destroy the enzyme. Maybe you can say that for these words, okay, these words, maybe you can add on. You write like this is not really wrong. Okay, you can get about 2 marks like that. But the question is 4 marks. Okay, so you should write another 2 sentences. So you can write out that the enzyme have a specific shape. Or you can say that enzyme have a complementary shape to its structure of substrate. Then since now the enzyme is being denatured, right? So the active site of enzyme will change after denaturation. So this will cause substrate unable to bind to active site of the enzyme. They are unable to bind to it. So there will be no enzyme substrate formation. And then because it's about the temperature, right? The factor is about the change in temperature. You increase 30 to 40 to 50 degrees Celsius. So the temperature will be related to the kinetic energy. So you should mention kinetic energy in your answer too. Okay. You can add on at here. At the temperature, which is beyond 50 degrees Celsius, it will cause the kinetic energy of the both enzyme and substrate molecule too high. So this will destroy their structure. Okay, so this will destroy their structure. Yeah, so that's the answer you can write. Substrate unable to bind to the enzyme and denaturation. Yeah, and about the kinetic energy too high. And then for this, outline the roles of the bile in the body's yeah, digestion of fat by the emulsification. Yes, correct. This one. Then allow for the enzyme for easier breakdown as a salt. Then waste excretion. I didn't understand about this sentence, but it's okay. But the upper sentence correct. The upper sentence correct. But emulsification about, you write like this, you can get about 1 mark. Okay, because you should describe in more detail what is meant by emulsification. It's a process which is involved in breaking down of large lipid droplet become small lipid droplet, right? So what should you do in this way? Because you want to increase, you should mention this one, increase the surface area for fat digestion. So once you increase the surface area for fat digestion, you're able to increase the rate of digestion of fat. Means you make the fat digestion become more faster. They're able to do it faster. And then secondly, you see the answer is about 4 marks. Then secondly, you should write about what? You should write about the second function of the bile is includes it able to neutralize the acidic kind, right? Yeah, they're able to neutralize the acidic kind from the stomach in the intestine. This is because that once the stomach content reach the intestine, it will become very acidic, right? So those will be neutralized by the bile. Okay, so this is to increase the pH of the intestine content for pancreatic or intestinal enzyme to perform their actions. This is because that our pancreatic enzyme and the intestinal enzyme, they are prefer alkaline, slightly alkaline environment, right? Which is around the pH 8 to 9. Yeah, 8 to 9. Okay, so those are the answer. Neutralize, increase the pH of the content. And then for this one, yes, the answer is duodenum. Active transport different from the diffusion. Yes, you write about keyword require the energy, or you can add on require the ATP energy, and then against the concentration gradients. And also this one, active transport specific molecule. Actually, it's not only the active transport specific molecule, but the other process such as the osmosis. Osmosis only transport water, right? Water is also a kind of specific molecule. So this one is not really accurate to be included in your answer. This one. Okay, so you can change into another point. You should state that active transport, they require carrier proteins in the membrane to help them to transport specific molecule. So the keyword will be this one, carrier proteins. Okay. And then this one, important of the microbiology, they give you 2 marks. So you should write that the important is you're able to increase the surface areas and you're able to increase the rate of the nutrient absorptions. The second point, to increase the rate of the nutrients absorption. Yeah, so every time when you want to when you want to write the answer, please read the question and also look at the answer. Look at the mark. How many mark will given for this answer? Then you should write according to the mark. If they give you about 2 mark, you just write about 2-3 sentences only. If they give you about 4 mark, 5 mark, then you should write more. If you write more, you're able to hit the target 3 point. But you should be confident. Okay, yes, this one is mucus. So the mucus function is they protect the harmful substance like bacteria. Okay, I think you are like missing every point together. You write until here is enough already. It means that you protect your digestive tract from the harmful substance like any pathogens. But this one, enzyme and acid, you should separate into another point. So you can see my way of writing will be like, yes, to protect the intestine from the pathogen attack. Then secondly, to prevent self-digestion by the digestive enzyme. Self-digestions, such as, for example, maybe such as pepsin in the stomach. Because your stomach content is very highly acidic, right? So possible they might be able to corrode your stomach lining. They might be able to corrode your inner stomach lining. So that's why your stomach lining, they need to cover with very thick layer of the mucus to prevent self-digestion happen by the digestive enzyme. So this is the second point. Then the third point will be to prevent physical damage by the food passing through intestine. Okay, so for this one, I can give you about 1 mark because I saw, protect against harmful substance. So this is one of the point. Then for this one, lactose. Where is lactose? Okay, lactose is not really involved in this one, nutrient absorption and transportation antigens. This is not really clear. And then about these nutrient absorptions, okay, what kind of nutrient will be absorbed by lactose? You have 3 kind of nutrient, right? You have glucose, you have amino acids, you have fats. So what kind of nutrients able to enter into lactose? So you should write your answer in a specific way. You should write out that the lactose, they will absorb the fatty acid and glycerol. The role is to help to absorb fatty acid and glycerol. Okay, this is first mark. Then secondly, you can write that lactose help to transport the fatty acid or fat into the lymphatic vessel. I'm not sure you still remember that. Last time I got include the point in the human digestion note. Yeah, the point, which is this one, which is about the pathway for the lactose and the glucose and amino acids. So for the lactose, it will be another pathway, which is this pathway they include in the lactose system. For example, I share to you lactose system. Oh, this one. Oh, sorry. Okay. I think you can see my screen, right? This pathway, this is a pathway here. This one. The lactose, they will absorb the fatty acid glycerol and then they'll enter into the lymphatic vessel. So this is a point that you need to write. Okay, and then this start from here, you no need to write so much detail. Just need to write that the lacteal, the enter from the lacteal into the lymphatic vessel is enough for you already. But just for you to know that actually the pathway is from the lymphatic vessel, they will enter back into hearts, and then they will absorb by the body cell. Yeah. So we continue. Okay. Okay, this one. Gallbladder is an organ, correct? But endoplasmic reticulum is a cell structure. It's not tissue. It's not tissue. Intestine epithelium, they will be tissue, because intestine epithelium, it will be a group of tissue together. Okay, a group of tissues, they will be forming together. Then they will perform this epitheliums. So epithelium is a tissue. Intestine tissue, it refers to the intestine tissue, which is formed by the group of cell. So the cell is an intestine cell or intestine villi cell. Ileum is the organ. So this is the correct answer. And then for this one, okay, the answer is correct, just like the gum is a pronunciation arrows. Okay, breaking down of solid food and chew for the mechanical digestion, yeah, it's correct. And then for this one, how the tooth decay occur? Yeah, you say that the acid. How many marks they give you? They give you about 4 marks. 4 marks. So this is number 23. 4 marks. So you write about the bacteria stay in the mouth due to the food leftover. I mean the leftover. So they will produce the acid. So the acid will dissolve the animals. Yeah, 1 mark for it. Dentin is exposed and begin to dissolve, causing decay and physical discomfort. Okay, this one you might add on another point. The tooth decay at the end, the tooth decay might reach nerve ending. Leading to pain. Okay, so that's all for this. Yeah. Number 24 is, yeah, I get the correct answer. Mucus and the mitochondria, yes. The gastric juice and describe its functions. Okay, gastric juice, they contains the hydrochloric acids. And then hydrochloric acid help to dissolve food. Okay, hydrochloric acid is not able help to dissolve food. Please remember that. This is not correct. But this one is correct. Okay. They provide optimum pH for the pepsins, and they destroy the harmful bacteria. These two correct. And then you can add on hydrochloric acid, they're able to denature other enzyme of the microorganism. So this is how they're able to kill other pathogens. Okay, so that's all, I think. Yeah, mucus. Okay, emulsification, yes. It's number C. Then small intestine and the lacteal, yeah. Okay, finish. So we are finished discuss for this one. So you just need to write the specific point for your answer in order to get mark. So, okay, now for this one, okay, just draw some specific question only for this one. So this will be B. Okay, which diagram represent the action of lipase? Lipase help to perform lipid digestion, right? Lipid digestion, they will break down the lipid. So lipid is actually fat also. They are made out of the triglyceride. This one is triglyceride, this one. So triglyceride, they are actually made out of the 1 glycerol and 3 fatty acid. This is the 1 glycerol. And this is 3 fatty acid. So they will be break down by the lipase, which is an enzyme. So they will form like this. Okay, they can be separated in this way. This one and another 3. 1, 2, 3. So the answer will be boy. Okay, because this boy show that this is a lipid, right? This is a lipid. This is a lipid. Break down by this enzyme, they will form 3 fatty acid. This 1, 2, 3, it refer to the fatty acid. 脂肪酸 fatty acid. And then this will be the glycerol. Yeah. Okay. Yeah. And then another question. This one. Which structure is duodenum? I think you miss out this question. But the duodenum will be located at here. Here. Okay. Remember about the locations of each organs. Duodenum is located at here, which is the entry point into the small intestine. And then the below questions. Here, this one. So the answer will be A, okay? Since the small intestine, they will absorb more food than the Y. Because small intestine is the longest part than the large intestine. I mean, the length of the small intestine is longer than your large intestine. So that's why small intestine able to absorb more water compared to the large intestine. Even though the function of large intestine mainly is for the water and mineral ions absorption, but small intestine, they're still able to absorb more water. Okay, so see, small intestine is very functional, right? They're able to absorb the nutrients and also they're able to carry out the digestion, the chemical digestions. Yeah, I think that's all for the question. So this one, where does the most water absorption take place? So the answer will be in the small intestine. It will be here. Okay, so we finished discuss for this chapter 7, paper 2 and paper 4. So Ethan Suyang, may I ask, last week you got complete this question? This one, chapter 4, paper 4, part 1, past year. You got try out this? Because I didn't receive your homework. I'm not sure you got try out this or not.
I didn't finish.
Yes or no? Haven't, or both of you haven't tried or not?
Haven't finished yet.
Oh, haven't finished yet. Okay, it's okay. Okay, I think, okay, we may start on a new chapter if like this. Yeah, because I wait you finish first, then we can discuss your answer. Because from this way, you can learn about what is the correct way for writing the answer. Then please open your notes for the photosynthesis. Okay, you go back to try out the question again. Maybe we can start a new chapter, photosynthesis. So photosynthesis, I think I sent you by the WeChat already, right? This note. Do you receive it? The notes. So can I start now?
Yeah.
Okay, sure. Photosynthesis. I mean, this chapter we study about plant nutrition. Okay, because plant just like human, right? They also require the nutrition, but their nutrition is much more simpler than the humans. So now we talk about photosynthesis because plants is a autotrophic organisms. It's autotrophic. What does it mean by this? You learned about before, right? The heterotrophic nutrition and autotrophic nutrition. This one, autotrophic nutrition. This means the plants they're able to produce their own food. So in human or animal, we are under this heterotrophic nutrition because we need to depend on the external food source. we are unable to produce our own foods. So plant able to perform photosynthesis because they contain the chlorophyll. This one, chlorophyll. Yeah. So actually, what's mean by chlorophyll? I think you heard about chloroplast, right? In chapter 2, chloroplast is a kind of organelles which is found in that tree plant cell. I'm not sure you still remember or not. There's the 3 type of plant cell, they contain chloroplast. Chloroplast. So that 3 kind of plant cell is we know as the palisade mesophyll cell. This one, palisade mesophyll cell. And then the spongy mesophyll cell. And then another one is gut cell. So Adam, so tell me, these 3 cells, which cell contains most number of the chlorophyll? Which one? I think which cell contains most number of the chloroplast? Is palisade mesophyll cell or spongy or gut cell? Which one? Have a guess.
Palisade.
Palisade, correct. Yeah. Palisade they contain the most number of chloroplasts, then followed by the spongy, and then the least number is in the gut cell. Yes, because palisade mesophyll cell, they are located under the epidermis of the leaf. Yeah, so they can allow them to direct expose to the sunlight. So whenever we want to write a photosynthesis equations, during exam, okay, you cannot write this. You cannot write this. You should write in this way. You should write chemical formula. C6H12O6, okay, plus H2O. And then, yes, on top is light, below is chlorophyll. Then you release the glucose. Glucose will be this one, C6H12O6, and release the oxygen. And then please remember to balance your equations. Put 6. Like this way. Okay, so this is the chemical equation that you should write in your exam. If the question asks you in this way, state the balance chemical equation. So please remember that the below part is chlorophyll. It's not chloroplast. It's not chloroplast. Chloroplast. Okay, let me describe to you. Actually, the relationship between chlorophyll and the chloroplast is, chloroplast is a structure, right? It's an organelles like this. And then chloroplast is double membrane. They contain inner membrane and outer membrane, just like the inner membrane is normal. It's not highly folded like mitochondria. See, this is inner, this is outer. This is the inner membrane. And then the chloroplast, they contain the granum. We know as this one, granum. It's like this way. The pancake, I told you before. Like this. So actually, where is location of the chlorophyll? Okay, this is the, we call it as the granum. This is granum. Granum, they allow light reaction happen. Light reaction means that the reactions happen during daytime. And then we have the stroma at here. Stroma is a fluid. It's a fluid-like substance found within the chloroplast. Right. Okay, a lot of liquid here. Yeah. So stroma, actually, they carry out the dark reactions. Dark reactions means that the reaction happens during nighttime. So during night, okay, let me rephrase. During daytime, the plants, they will absorb the sunlight. Okay, in the daytime, plant absorb sunlight. Then, however, in the night time, plant will produce the glucose. They will synthesize the glucose, which is this one, glucose, C6H12O6. So that's why we know that the plants, they able to produce their own glucose. And usually where they produce the glucose in the leaf. So usually leaf, they will know as a source. Okay, you will learn about these two terms, source and the sink. Source. Source means that the place which they produce the nutrients. Sink means that the place which they store nutrients. Yeah, like this. Okay, so let me go back to the notes. Yeah, so what is photosynthesis? Photosynthesis is a process which is the plant make their own carbohydrates. The carbohydrates, they refer to the glucose here, C6H12O6, from the raw material. The raw material is they refer to this. Oh, sorry. I think I write the wrong thing. Oh my God, I did academy, sorry. Yeah, this one is carbon dioxide and the water. I just realized I write wrong. This one. Okay, so it's carbon dioxide and the water. So they will produce the glucose, C6H12O6, and the oxygen, O2. So the raw material they refer is the carbon dioxide and the water, these two raw material. Okay, so actually photosynthesis is a process which is they are converting the, we call it as the inorganic substance. Inorganic substance is they refer to this one, carbon dioxide and the water, they are inorganic. Inorganic, 无机. So they will convert become the glucose, which is organic. Which is the organic compound. So using the light energy. Yeah, so please remember about this equation. And then now we need to talk about the theory, some theory of the photosynthesis. So you can see that the plant, they will absorb the carbon dioxide. Okay, they absorb the carbon dioxide from the leaf stomata. Stomata is the small hole found in the lower epidermis of the leaf. And this stomata actually is formed out by a pair of the gut cell. A pair, this one, okay. A pair of the gut cell. So the carbon dioxide, they will enter into the plant through the stomata, which is a small hole under the leaf, under the lower epidermis of leaf, like this. This is a leaf, right? So below it, there will be the small hole, stomata. Stomata is usually at the lower epidermis. It will not be at upper epidermis, because upper epidermis, you have the cuticle and the upper epidermis cells. And the water, the plants, they will absorb the water from the roots. Yeah, from the roots. And then they will transport through the xylems to the leaf. Okay, the roots is about in this way, like this. This is a plant, right? And then this will be the roots. Okay, and then the roots contains the root hair cell. This is root, and then they contain the root hair cell, like this. Root hair cell. So this root hair cell, they will help to absorb water and mineral ions. Two things. One thing is water. Water, they will absorb by osmosis. And the mineral ion, they will absorb by active transport. So after that, the water pathway, you should know about the water pathway, is the water, they will absorb by the root hair cell, and then they will get into the, we call as in the xylem vessel. enter into xylem vessel. And this xylem vessel are found in the roots, stem, and also leaf. So they will travel up. The water, they will travel out, which is against the gravitational pull because they are being attracted by the force. Okay, this force we call as the transpirational pull. Transpirational pull. Transpirational pull. Is a kind of the pulling force. They help to attract the water up from the roots to the stem until they reach the leaf. So this is they will reach a leaf like here. This is a leaf, right? So what will they do is once the water, they will arrive in the leaf. And in the leaf, there's a lot of cell, right? They contain the placid mesophyll cell, spongy mesophyll cell, and then the gut cell. So the water, they will enter into those cells in the leaf, which is the mesophyll cell. Okay, so the way the water able to enter the cell is by osmosis again. And then from the cell, they will enter into the air space and then they will release the water through the stomata. So there will be water losing in form of water vapor from the stomata. Water loose in form of water vapor from stomata. So this is we know as the transpiration. Transpiration is a form of the water loss. Transpiration. 水从这个叶子的stomata蒸发 transpiration. It's kind of evaporation, but you cannot write evaporations. You should write transpiration. And then continue to say that glucose, they are being made in the leaf. Because leaf contain the chlorophyll. So glucose are being made by the leaf. And then glucose, they will be become what? They will become the sucrose. Later. So after the leaf produce the glucose, right? So the glucose, they will be actively transport into the phloem and then they will become sucrose. Which is another form of the disaccharide. Actually, there's a reason that the plant need to transport in form of sucrose but not glucose, because when they transport in form of this sucrose, because sucrose is disaccharide, right? It's a double glucose molecule, disaccharide. But glucose is monosaccharide. So sucrose is very stable. It's not really affected the osmotic pressures of the plants. So that they will prefer transport in form of sucrose, even though during photosynthesis they produce the glucose, but after that they will become the sucrose. So sucrose, they will be used in the translocations. Translocation, this process. I think you heard about this before. Translocation is happen in the phloem. So generally, glucose, they are actually needed by the plant because they also, the glucose are needed by the plants to perform the respirations to release the energy. Even the plants, they also able to do the respiration. Plant able to do these two process, photosynthesis and the respiration. Plant usually, they will do the photosynthesis in the daytime. In the daytime, because there is a sunlight. There's a sunlight. So the sunlight, they can be absorbed by the chlorophyll in the leaf. So plant do the photosynthesis in the daytime. However, in the nighttime, plant, they will switch, become the respiration. So they will become similar like humans, okay? Because respiration is I mean, the equation is totally in the opposite way, right? Which is they involve in the glucose and the oxygens. Involved in the glucose and oxygens, and they will be converts, become the water, carbon dioxide, and then they will release the energy. So that's why there's a story say that the plant will compete your oxygens during the nighttime. Because it's this purpose. So okay, after the plants, they will produce the glucose in the leaf. So the plant, they will also able to release the oxygens. They release the oxygen throughout the leaf through the stomata. The oxygen is being produced during the photosynthesis as the byproduct. Actually, oxygen is kind of byproduct. So the oxygen, they will be released out to the environment. So the oxygen, they will be used up by the human being or the animals to perform the cellular aerobic respirations. So this is the cycle. Okay, so now let's look about some photosynthesis theory. So basically, the photosynthesis is which the light energy able convert into chemical energy, because you see from the inorganic become organic. So in the bonds, they are holding the atoms in the glucose molecule together. So they will produce the glucose. So the glucose contain the chemical energy. So the product of photosynthesis is they will produce a glucose. So glucose, they will be used become the source of energy in the respiration. After that, the glucose, they will convert into the starch for storage and into lipids for energy source into seeds. So they also able to convert become the cellulose and then become the amino acids to use to make the proteins. So you can see this mind map. This is a glucose produced by the plants. So after that, they will convert become the sucrose for transport. See, sucrose for transport during the translocation process. In the phylum, translocation in the phylum, and then glucose, they will be used up during the cellular aerobic respirations, especially in the night. Okay, because plant also able to perform the respirations. So this is to release energy for the cell, and also glucose able to make up others polymer, okay, like starch or cellulose, because starch and cellulose, they are polysaccharide, right? They are the complex form of the carbohydrates, which is the polysaccharide. So glucose able to combine together. So this process we know as a condensation, which is they will combine hundreds units of glucose together, condensations. So they're able to produce a polymer like starch and cellulose. So the starch is generally used for storage purpose in the plant. Okay. So this means that the plants, they will store their excess energy in form of starch. I mean excess glucose, sorry. Excess glucose in form of starch. However, in human, we will store excess glucose in form of the glycogens. So this is the difference between them. Then you have the cellulose to increase the strength of the cell wall. So the cellulose also made up from the glucose. And then you're able to convert the glucose, become the fats and oils for the energy storage in the seed. So this is function for the glucose in the plant. Okay, so you still remember about this. This is the required as the polysaccharide that you learned in chapter 4. See, this is starch, glycogens, and the cellulose. Then you need to know about the structure, shape, because they might be come out in the paper form. So for the starch, it will be less branched compared to glycogen. Glycogen, they will always define it as a highly branched molecule. Highly branched. Because there's a lot of small branch, right, like this, which is from the middle part. This is a starch. And then this is cellulose. Cellulose is not branching molecule, but cellulose is like this. Okay, so the cellulose make the cell wall become very rigid. They'll form a rigid cell wall. Okay, and then this kind of question, they will like to coming out in the paper 6, which is experiment questions. So they will design some experiments, then they will need you to investigate about the photosynthesis. Okay, so you can see that although the plants make the glucose in the photosynthesis, but the leaf cannot be tested for the presence as the glucose is quickly used. So they will be converted into the substance and transport as a starch. So you can see that the glucose in the leaf, usually they are exist in form of starch, because starch is a kind of energy storage molecule, just like in the glycogen. So starch is stored in the chloroplast. So in the chloroplast, we can find starch. Usually, we're able to find the starch from the granum. It's not, it's a stroma. Here. Usually, we're able to find the starch in this place, which is a stroma. We're able to find some like starch grain at here. For example, starch grain, starch granule. Because stroma, they carry out the Dart reactions, right? Which is they will produce the glucose. You will learn this later when you're going to learning about higher level of biology. Okay, so they did an experiment, which is they want to check for the presence of the starch inside the leaf. So initially, they will boil the leaf with the boiling water because they need to kill and break down the cell first. So and then they might ask you, what is the purpose that you need to boil the leaf with the boiling water? So this is your answer. You need to break down the cell wall. So why you need to break down cell wall? Because you need to allow some substance found inside the leaf to be released out, right? So you can see after that, leaf is hit with the hot ethanol. And then this ethanol able to remove the chlorophyll. So the color change for the iodine can be seen more clearly. And then the leaf is spread out in the white towel and covered with the iodine solution. So now they use the iodine solution because they want to test for the presence of starch in the leaf. So in the green leaf, the entire leaf able to turn blue-black if the photosynthesis happen in all area of the leaf. So, but there might be some area on the leaf which they do not contain any chloroplast or chlorophyll, right? So when the leaf is tested, only the area contain the chlorophyll, they will stain blue-black. So other place which they do not contain the chlorophyll, they will not stain as blue-black. Yeah. So yeah, it's something like this. Okay, first you need to boil the leaf in the boiling water, and then you need to soak the leaf in the ethanol. So the ethanol, they able to dissolve away the chlorophyll. Yeah, see, you need to remove the chlorophyll by the ethanol. So you can check about the presence of the iodine, able to stain the starch later. Okay, so something is like this. So this is before the starch test. So you can see this is a green area, right? This is green area. Then some area, there will be yellow area. So this yellow area means that they do not contain chloroplast. So only this green area, they do contain the chloroplast. So when they are being stained with the starch solutions, oh, sorry, when they are being stained with the iodine solution, this one iodine solution. So the iodine able to detect the starch presence in the green area because they contain the chlorophyll. So they were able to change blue-black. So the regions which is they did not contain the chloroplast, they will not change blue-black. Yeah, so it's a very simple experiment just to show you that. Yeah, like this. So only the area they contain the chlorophyll, they will change blue-blacks. Show the presence of starch. Okay, and then some care or precaution that they must be taken while carrying out this practical is because you're playing with ethanol. Ethanol is a kind of the highly flammable chemical reagent, right? So you might easily to burn off. So that's why you need to take care of it. Okay, so to heat the ethanol in is the electric water bath rather than using the beaker over the Bunsen burner. So this is whenever they ask you in this paper 6, which is about the experiment questions, so you can write. It's okay. I will show you example of question in next class. And then this one, the need for the light in photosynthesis. Then you need to explain that. Okay, this one, the need for the light. So before starting the experiment, the plant need to be discharged, which is you need to remove away the starch by placing in the dark cupboard for about 24 hours. So this to ensure that any starch already present in the leaf will be used up and will not affect. So, and then after you discharge the leaf, you need to cover with the aluminum foil and place in the sunlight for days. So after that, the leaf can be removed and test for the starch using the iodines. So only the area they cover with the aluminum foil will remain as the orange brown as it not receive any sunlight, this one, and could not able to perform the photosynthesis. So only the area exposed to sunlight, they will change blue-blacks. So this is they play second experiment, which is they cover some area of the leaf with the aluminum foil. So they want to prevent the photosynthesis happen. So if there is no photosynthesis happen, there will be no starch formations. So they cannot be detected by the iodines. So this shows that the need for light in the photosynthesis. And then how about the carbon dioxide? So the carbon dioxide is actually exists in our atmosphere. Okay, but whenever you want to do the experiment, test about the presence of the carbon dioxide concentration in the environments. Yeah, so this is how you should need to do. Prepare the bag containing the sodium hydroxide. So this sodium hydroxide is a chemical reagent. They're able to absorb the carbon dioxide from the air around one leaf. Okay, and then you tie a clear bag with the water, so we should not able to absorb the carbon dioxide. You see, only sodium hydroxide able to absorb carbon dioxide. Water cannot. And then you place the plant in the bright light for about several hours because you need to perform the photosynthesis. So only the leaf from the bag containing the sodium hydroxide will remain as orange brown, as it could not photosynthesize due to lack of carbon dioxide. Why? Because that the bag contains the sodium hydroxide. So the sodium hydroxide already absorbed the surrounding carbon dioxide. So this means that the leaf unable to absorb any carbon dioxide. If they cannot absorb any carbon dioxide, they will not perform photosynthesis. So this is the answer. Then how about the rate of the photosynthesis using the water plant? Okay, this is the rate of photosynthesis. It means that they need to calculate. Okay, this can be, perform some calculations to check whether the photosynthesis for this case is higher or lower by calculating the number of bubble produced. So actually this number of bubble is they are calculating the oxygens, because oxygen will be released from the water in form of bubble. So initially they will choose two plants, which is the Elodea or Camboba, just choose either one. So after that, if there's a photosynthesis occurs, they will produce a bubble oxygen gas released in the water. So what will happen is, you can see, this is very common experiments asking in the exam. So this is a plant, right? So, and with the, we call as the boiling tube, with the inverted boiling tube. So they will calculate the how many gas bubble released from the boiling tube. Released to the boiling tube from the plant itself. So if there's more gas bubble being released, that means there will be highest rate of the photosynthesis. And then they will change about the position of the lamps. This one, the position of the lamp. So whether it's become nearer to the plants or further away from the plants. Okay, so this is effect of changing light intensity on the rate of photosynthesis. They alter the changing in the position, they will alter the photosynthesis rate. So if the light intensity stay more clear, stay more near to the plants, they able to increase the rate of photosynthesis. So there will be more bubble produced. So if the plants stay further away, so this will able to reduce the rate of photosynthesis. So there will be less bubble produced. So that's all. Okay. Yeah. Okay. Let me explain to you about some exam tips. The way of measuring the gas given off in this experiment, we're able to measure the volume of gas produced using inverted measuring cylinder with the radiation filled with the water. This will be another way of measuring how much oxygen gas we produce during the experiment. Yeah. Okay, now let's talk about the limiting factor. So actually, what's limiting factor? So from those experiments, which is the light, you study about light intensity, how light intensity able to affect the rate of photosynthesis. Then secondly, you study about how much is we call as the carbon dioxide, the absent and present of carbon dioxide. And also you're able to study the carbon dioxide concentrations. If you have a high concentration of carbon dioxide, you have the higher rate of photosynthesis. Higher concentration of carbon dioxide able to increase rate of photosynthesis. And also you're able to study about the presence of light. Yeah. So this shows you that the photosynthesis rate is being affected by these three main factors, which is temperature, light intensity, and the carbon dioxide concentrations. The photosynthesis is being affected by the temperature is because that photosynthesis is being catalyzed by the enzyme also. This one, enzyme. Just in the IGCSE, they did not ask you to study about what type of enzyme to control the photosynthesis, but it's a enzyme catalyzed reactions. Photosynthesis is a kind of enzyme catalyzed reactions. So the temperature able to affect the enzyme structure. Okay, so you can see whenever you increase the temperatures, so the rate of photosynthesis, they will be affected. So they will produce a graph just like the enzyme graph with we call as the low temperature, optimal temperature, and the too high temperature until the enzyme is being denatured. And this will cause the decrease in the photosynthesis rate. Yeah. Okay, so just similar to enzyme, which is whenever you increase the temperature, you're able to increase the rate of photosynthesis. As you're able to increase the kinetic energy, right? You're able to increase this one, increase the kinetic energy of both substrate and enzyme. So after you increase the kinetic energy, you're able to form what? You're able to form the enzyme substrate complex. Increase formation of the enzyme substrate complex. Sorry, before that, we should write on this one. Actually, you are increase the frequency of collisions. You increase the collision between enzyme and substrate. So you're able to increase formation of the enzyme substrate complex. Yeah. So after they reach the optimal temperature, which is they get the highest rate of photosynthesis, but beyond that, the too extreme high temperature, they will denature the enzyme again. So now the substrate unable to bind to the enzyme active site. So this cause the rate decrease very fast, decrease rapidly. You can see until become no activity, no enzymatic activity. Okay, so you see the diffusion is just same like you're writing for the enzyme. And how about light intensity? More light, faster rate of photosynthesis. Okay, but there is a limiting point. So you can see this graph. Whenever you increase the light intensity, you are increasing, right? The rate of photosynthesis will increase, but until they reach a point. Because when they reach this point, the limiting factor will become other. It's not light intensity. Maybe it's due to the carbon dioxide concentrations, or maybe it's due to the temperature itself. So if you want to further increase the graph become like this, you need to increase other factors. Like you need to increase temperature, or you need to increase the carbon dioxide concentrations. If not that, you further increase light intensity also become not really helpful, because after that, once they reach the limiting factors, okay, means that means the light intensity unable to further increase the rate of photosynthesis, they will just remain constant like this way. Okay, see, increasing the light intensity until they are limiting by other factor. Okay, so how you able to expand this? See, at low light intensity, increasing the intensity will increase the rate of photosynthesis. So at a certain point, increasing the light intensity able to stop increasing the rate. So now the rate will become constant regardless how much light intensity increase, because other factors become limiting the rates. Okay, so it's temperature or carbon dioxide. Then how about in the carbon dioxide concentrations? So the graph is just same to the light intensity just now. It's just same like this, right? Which is whenever the carbon dioxide they reach a certain point, limiting point, so it will be limiting, will be caused by other limiting factors such as the light intensity or the temperature. Okay, see, the trend will continue until some other factors required for photosynthesis. They will prevent the rate from increasing further because it is now in short supply. So actually they are referring to either the temperature is not high enough or there's not enough light. Okay, that's all. So remember about these three factors they will affect the photosynthesis rates, which is the light, the temperature, and the carbon dioxide concentrations. So the temperature is just similar with the enzyme catalyzed reactions. Then how about we talk about in the glass house? Glass house is something like this one. Glass house means that you want to create environments which is suitable for some certain plant. So how to create a glass house? So you should know about the limiting factors and how they able to affect the rate of photosynthesis. So you're able to control the factor in the glass house to ensure the maximum crop yield for the farmers. Actually, this glass house is very common in the temperate country, which is they got 4 seasons, which is winter, summers, spring. Very common in that country because especially in the country which we have a very long winters and long autumns, because winter and this autumns is not really favorable season for the plant to grow, right? Because winter they will be lacking of the sunlight. So this is the reason that they need to create a glass house, which is they create an artificial design conditions for the plant to grow with a maximum exposures to the sunlight to increase their rate of photosynthesis. So when you want to design your glass house, what kind of equipment that you need to prepare? You need to prepare about the artificial heating. You need to give some heat, right? Because you need to allow your photosynthesis able to carry out in the optimum temperature conditions. See, enzyme controlling the photosynthesis can work faster in the slightly higher temperature, just slightly higher temperature, not too high, not to destroy the whole enzyme. And then you need some light, artificial light. So you're able to allow your plant to carry photosynthesis for a longer time. And then you need to increasing the carbon dioxide content for the light inside. Okay, actually you can adjust your carbon dioxide content. You can become very low content or become very high contents. And then you need the watering. Okay, so nowadays the agriculture is very modern and advanced. So they will be automatically watering plants designed at that. So whenever there's a timing, so the water will be released out to the plant. Yeah, that's all. Okay. In tropical country where the temperature is much more hotter, glass house may still be used to control other conditions, but they need to ventilate to release hot air and avoid temperature rising too high. This temperate country is not temperate, sorry, it's tropical country. Tropical country just like in the country with very hot seasons. Okay, in the very hot season, just like in the Malaysia, in my country, it's very hot. So such a hot season, right? So the hot season, they're able to affect the growth of the plants because plant cannot be grow in the condition which is too hot. So you need to reduce down the temperature. So that's why you need to add on additional fans. You can add on like this ventilations by add on the additional fan. So this able to help you to reduce the housing temperature. Okay. Okay, so now let's go to 6.4. This plant nutrition is very simple chapter. You just need to know about there's 3 factors only. And then now we talk about this investigations of the gas exchange. So what is the effect of light on the gas exchange is, you can see this one. Actually, this shows you the relationship between the respiration and the photosynthesis. This means that in the day, whenever the sun is bright, the plant, they will carry out the photosynthesis in a faster rate than the respiring. Okay, this shows that the plant actually able to carry out 2 processes. One is photosynthesis, one is respiration rate. Just like which one is higher rate than other. So during daytime, during daytime, plant usually they will do the photosynthesis more. They do more photosynthesis, right? So they do less respirations because during daytime they have the light. And also they have the carbon dioxide source, enable them to do the photosynthesis. So you can see there is a net intake of the carbon dioxide and the net output of the oxygens, because they absorb carbon dioxide and they release the oxygens in the daytime. So we can investigate the effect of light on the net of gas exchange in the aquatic plants using the pH indicator, such as the hydrogen carbonate indicator. This hydrogen carbonate indicator is very common question to ask in the paper 4 and paper 6. Okay, hydrogen carbonate indicator is a kind of solution test for the presence of the carbon dioxide. They test for the carbon dioxide presence. So if there is a presence of carbon dioxide, this will cause the indicator change become which color? They will change become yellow or orange like this. If there is a increasing carbon dioxide concentrations. Carbon dioxide concentration. So the the color become more yellow orange. If there's less carbon dioxide, the color will become more pink and purple. Okay, so this is by using this indicator, we're able to investigate the effect of light on the net of gas exchange, because carbon dioxide is an acidic gas. Please remember that carbon dioxide is an acidic gas, yeah, when they dissolve in water. Okay, so they're able to detect by the hydrogen carbonate indicator. Okay, yeah, this picture just show you that photosynthesis only occur in the chloroplast found in the tree cell. Yeah, I told you just now. So in the daytime, they require the light for the photosynthesis, but temperature is also very important. So temperature cannot be too high. Okay, I think that's a point. Plants also photosynthesis during the daylight hours, which they need to take in the carbon dioxide, and they release the oxygens made in the photosynthesis. So during nighttime, okay, sometimes student might ask you, me, does plant carry out photosynthesis in the nighttime? The answer is no, because in the nighttime, there's no presence of light. If there's no light, plant are unable to convert the light energy become the chemical energy. So plants do not perform photosynthesis in the nighttime, but they will do respirations. So during nighttime, they will switch become respiration mode, which is they will use up the glucose and the oxygens and release the carbon dioxide. Water, just like in the human do. So they will take in the oxygens and then they will release the carbon dioxide. So this shows that during daytime, the photosynthesis rate is higher than respiration rate. However, during nighttime, the respiration rate will be higher than the photosynthesis rate. Because definitely there's no photosynthesis happen in the nighttime. Okay, so this indicator they show you the carbon dioxide concentrations. They show you the concentrations. You can see that if you have the highest rate of carbon dioxide, change become yellow. If your lowest rate, if there's no presence of carbon dioxide, they become purple. Okay, so if there is the increasing carbon dioxide concentration, that means that the respiration rate is higher than photosynthesis rate here. Because respiration rate, they release the carbon dioxide. Photosynthesis, they consume the carbon dioxide. So once respiration rates, they release the carbon dioxide, it will cause the excessive accumulation of the carbon dioxide in the atmosphere. So they can be detected by the indicator. So once the photosynthesis rate more than respiration rates, photosynthesis, they will be use up the carbon dioxide. So there will be lower concentration of carbon dioxide available in the surrounding. So this indicator just allow you to check which process happened in the higher rate, either it's respiration or in the photosynthesis. So please remember about this process. And then this one, this is 3 tubes with the leaf content inside. And then they are given with the conditions, either it's light, dark. Okay, you can see this one, A and B. The leaf given in the light conditions, right? So the indicator, they change into purple because the photosynthesis happens. They use up already the carbon dioxide. Now, if the leaf are put into dark conditions without any light, so they will switch, become respiration. So they will use up the carbon dioxide. They will release the carbon dioxide. They will release carbon dioxide. Actually, right here, release the carbon dioxide. For this one, A, they will use up the carbon dioxide. Okay, that's all. And then now we talk about the last part, structure of the leaf. I think this one is last one. Structure of leaf. We will finish chapter 6 for today. I hope that it's not too fast for you. Is it okay for you, Ethan Suyang? Can you get me? Can you follow me?
Yeah, kind of.
Oh, okay, hopefully. I'll give you exercise later. Now we talk about structure of the leaf. So leaf is a source which they produce glucose. Leaf, they have upper epidermis at here, and they have the lower epidermis at here. Okay, and then please note that this upper epidermis, they are being covered by the layer of the cuticle. It's not shown in this picture, but I think you need to know cuticle. Cuticle is kind of waxy layer. Cuticle. Okay, it's a transparent waxy layer. Transparent waxy layer. It's very transparent, so they able to light just directly penetrates into the mesophyll cell, especially the palisade mesophyll cell. So the plant, they have these 3 important cells, which is the palisade mesophyll cell, and so the spongy mesophyll cell, and then the gut cell. This 3 cells is so important. And then you need to know about the structure of these 3 cells. Later I describe to you. And then these 3 cells, you can see that the orientation for the chloroplast, right? Like this one, chloroplast at here. This is chloroplast. And then at the spongy mesophyll tissue or spongy mesophyll cell, you can see there's a lot of space, right? See, this is space, this is space. Wow, so many space. So this space we call as air space. Air space just contains only air. There's no, I mean, they does not contain any water. They just contain only the air. And then at the layer for the spongy mesophyll tissue, there's a kind of the specialized tissue we call as a vascular bundle. This one. Please highlight this one. Vascular bundle, this one. Okay, vascular bundle. Let me show you a true picture. Vascular bundle. Oh, this one. Vascular bundle, they contains two things. They contain xylem and phloem. And then you need to know about orientations for the vascular bundle. I think this picture is better for you. Xylem, phloem. Okay, I think I better show you this picture. So yeah, let me find a better picture to show you. Okay. I show you this picture. A little bit blurry, but you can see it. This bundle of tissue, they know as vascular bundle. And actually it's comprised of two cells. It's not two cells, it's two tissue, because xylem and phloem is tissue. So see, xylem and phloem, they are tissue found in the vascular bundle. They are referred to this structure, this circle, this whole thing. This is vascular bundle. And then these vascular bundles, sometimes the questions, they will ask you to label which is xylem, which is phloem. So you can have a look. This is a xylem. This whole thing is vascular bundle, right? This whole thing is a vascular bundle. And then this is the xylem. And then this is a phloem. This is a phloem. Xylem and phloem. Okay, how to differentiate is xylem or phloem? Just look at the whole, the size of the whole. The xylem, they contain the largest size of the whole compared to phloem. Xylem, they contain larger side of the whole. Okay, phloem, they contain the smaller side of the whole. They always appear in this way. When you see the cell with larger side, that one is xylem. Smaller is phloem. That's all. And then you can see the stomata at below it here. A small hole known as stomata, which is at here. This is the stomata here. You can help me to label this hole. This hole is known as stomata. Because this stomata is formed by a pair of gut cells. It will be in this way. Whenever I need to draw a stomata, I just need to draw like this. Okay. And then this is a gut cell with the nucleus, with the vacuole. Because it's plant cell, because they contain the vacuole, so they contain vacuole. And also there are some chloroplast. This nucleus and also the vacuole and some chloroplast. So this two cell is known as gut cell. And then this hole is known as a stomata or stoma. Yeah, I think that's all for you. So this is, they show you how plant cell look like. This is plant cell. So the plant cell, they condense, just that you learned before, they contain vacuole, they contain nucleus, they contain chloroplast, and also they contain mitochondria. They have mitochondria, and also they have the ribosome. This small circle-like structure is a ribosome. So only these three cells contain chloroplast. Other cells they don't have. See, xylem, phloem, and the epidermis cell, root hair cell, they do not contain chloroplast. So they are able to carry out the photosynthesis. Okay. This shows that how the carbon dioxide enter into the leaf. How they do the leaf. So they will enter by the stomata, the hole here. Okay, so you can imagine that this stomata is just like your nostril. You have a nostril, right? 你的鼻孔. So the nostril will always open to allow the oxygen inhale and exhalation of carbon dioxide. This is for you. But in plant, do in opposite way. The stomata able to open and close. When they open, they able to allow the carbon dioxide diffuse inside. You can write down this. When the stomata open, they able to allow the carbon dioxide diffuse into the airspace. Because this airspace is located in the spongy mesophyll cell. Located into airspace of the spongy mesophyll cell. This is when stomata open. So how about when stomata close? When stomata close is when the environment for the plant is too hot, excessively hot environments. So this will cause the water loss, excessive water loss. From the plant cell through stomata. You can imagine, right? Whenever the plant is being placed in a very hot environment means that the temperature is so hot, and then there will be very low humidities. Means that high temperature, and then low humidity. Low humidity means that the air moisture is too low. 空气的湿度很少，很不够。So this will force the stomata close. Why they close? Because they need to reduce water loss from the plant. Yeah, so this is a reason. This is a reason that why the stomata open, why stomata close. They open to get in the carbon dioxide, to intake the carbon dioxide for photosynthesis. They close to prevent any further water loss during hot environments, when they are in the hot environments. Okay, so let's go back to this picture. Why the carbon dioxide from the surrounding atmosphere able to diffuse into the stomata? It's because that there's a higher concentrations in the carbon dioxide in the atmosphere compared to inside. Means that outside there are higher carbon dioxide concentrations, but inside have lower concentrations. So carbon dioxide able to diffuse into the leaf through the opening stomata into the airspace of the spongy mesophyll cell at here. This place, this is known as the airspace. After that, the carbon dioxide they will diffuse through the cell wall or the cell membranes of the mesophyll cell. So they will get into the chloroplast. So they enter the chloroplast, so they're able to perform the photosynthesis. Like this at here. Okay, so after that, the carbon dioxide they will be become the glucose and being produced in the stroma of the chloroplast. So the story is like this. See, from the atmosphere into the airspace. See, airspace in the spongy mesophyll cell. Because only spongy mesophyll cell, they have the airspace. They have the airspace. And then get into mesophyll cell and get into chloroplast. So they're able to do the photosynthesis. So that's the story for this leaf to get the carbon dioxide. Then we should study about the structure of the leaf. This is structure of the leaf. Okay, let's go with the picture. This wax cuticle. So this wax cuticle is placed on the upper epidermis of the leaf, which is just what I mentioned to you, around at here. This area, this one, at here, here. So it's a protective layer on top of the leaf, which is able to prevent water from the evaporating. Then upper epidermis is just very thin and transparent. Epidermis is a transparent cell because they need to allow the light to directly enter into palisade mesophyll cell underneath it. And then the palisade mesophyll cell is a columnar-shaped cell. Okay, please remember about the structure of this three cells. See, it's a columnar-shaped cell. Why? Because you can see that palisade mesophyll cell, they are in this shape, right? Tall columnar means it's a long rectangular shape. long rectangular cell. So it's tightly packed with the chloroplast to absorb more light to increase the rate of photosynthesis. So you can see that this plasmid cell, their orientation is they are very tightly packed. See, very closely packed to each other without any space, no space. It's not like spongy medulla cell. It's very loosely arranged with a very large intercellular air space. So you can describe it this way. Spongy medulla cell, okay, maybe I can write here. I write here. It's a loosely arranged with large intercellular air space. So actually, what does spongy medulla cell do? They contain the chloroplast. Yes, they're able to do the photosynthesis, but their functions actually is they need to perform the gaseous exchange. See, they contain the internal air space to increase the surface area for the gas diffusions, mainly carbon dioxide. They're actually for the gaseous exchange. Maybe I can show a picture, which is that how the leaf able to do the gaseous exchange. Leaf do gaseous exchange is by this. Okay, this is a very good picture. They show about how leaf able to do gaseous exchange. You can see from this picture. This is a stomata, which is a pore, right? So they allow the carbon dioxide enters, and they allow the oxygen to release from the stomata. And also, whenever there's a water loss from the stomata in form of the transpirations, they also go out from the stomata. I mean, the water, they will be evaporates, become the water vapor and release away from the stomata. So this water vapor is during the transpiration. Water vapors during transpiration. So transpiration means that involved in losing of the water in form of water vapor from the stomata. Okay, and then the lower epidermis involved in the gut cell and the stomata. So the gut cell is what they do. They're able to open and close. So they will absorb and lose the water. They will be losing the water to open and close. So when they open, they allow carbon dioxide to diffuse in and oxygen to diffuse out. Then for the stomata, is when the gas exchange takes place, they open during the day and close during the night. Stomata is a hole. This hole, whether they want to open and close, is being controlled by the gut cell. This gut cell. Okay, controlled by gut cell. So that's why the function of gut cell is they are help to control opening and closing of the stomata. These functions. So how they help in the closing and opening of stomata is they will either absorb or lose water. This means that whenever the stomata opens like this, this is how they open, right? When they're able to open, it's because that they absorb water. Water go inside. Water go inside. Water store in the vacuole. This is vacuole. Means that there will be large amount of water stored in the vacuole of the plant cell, of the gut cell. So they will become turgids. So they will expand inside. So whenever the gut cell, they lose the water, they will close. Like this. The vacuole will be shrinking. This vacuole will be shrinking. Actually, they are losing water. So they will be closed. So this is known as a flaccid gut cell. Flaccid. Because they lose water. And then these stomata, they allow the evaporation of water take place, which is the transpirations. Transpiration is a kind of the evaporation of water. Transpiration. Okay, then the bacial bundle, they contain xylem and phloem. So what does xylem do? Xylem, they will help into transport water and mineral ions, which is from the roots into the leaf to use for the photosynthesis and the transpirations. And the phloems, they will help to transport this one, sucrose and amino acid around the plants. See, this is sucrose, this is not glucose. Okay, so actually just like xylem is like drinking water. Plants cannot drink water like human, right? But they contain xylem, help them to absorb water and mineral iron for the usage by the plants. And this phloem, just like the humans eating the food, plant cannot directly eat food from the just like human, right? So the phloems, they will help to transport the glucose produced by the plant itself and then convert become the sucrose. So then the plant able to use those sucrose to do their things. So one is transpiration, one is translocations. Xylem is about transpiration. Phloem is about the translocation process. These two process. Okay, so I think that's all for today. Okay, we still have a little more topic to discuss, but it's just very simple only. Okay, I'll give you some questions to do, and please remember to attempt the chapter 4 paper 4. Yeah, this one, chapter 4 paper 4. And then I will see you in next class. Thank you, Adam and Suyang, for today. Do you have any question to ask me currently? Okay, thank you. Okay, thank you for homework. Just do more because your exam will be in the next year. Yeah, next year in 2026. And then we still need to practice some paper 6. I think you haven't attended paper 6, right? Paper 6, which is the experimental paper for the IGCSE. It's okay. We will do next time. Okay, see you again. Bye.